Good day, ladies and gentlemen. This little presentation is going to be dealing with how a nerve generates an action potential, which is basically a nerve impulse. And to begin with, we're going to look at a couple of important terms here and characteristics of nervous tissue to begin with. Okay, neurons, have several properties. First one is irritability. And besides neurons, also muscle tissue to an extent is irritable. But irritability simply means this the ability to respond to some stimulus. And in this case, the stimulus is going to be anything from a flash of light to a sound to a pinprick. But the nerve impulse can actually respond to this. And then the second property you need to talk about, another term, is conductivity. Conductivity is simply the ability to transmit that impulse or move it from point A to point B. Now, as we begin this discussion here, we're talking about the plasma membrane as being polarized. A polarized plasma membrane is one that is at rest. It's not going to conduct an impulse. Here's what that means. In this picture here, we've got a plasma membrane right here, and we have sodium and potassium ion channels here. So we're allowing some ions to pass through this. ECF, extracellular fluid, ICF, intracellular fluid. So this is on the inside. If you look at this picture carefully, you notice that there's lots of ions in particular here, but the ones that we really need to pay attention to are sodium and potassium. Notice that on the outside of the cell, the sodium has a much higher concentration outside than inside. But the opposite is true for potassium. Potassium has a much higher concentration inside than out. And this is a polarized membrane. Now, this difference is great enough that when you look at the number of positive ions, there's much more positive ions in the extracellular fluid than there is in the intracellular fluid. Okay, so in other words, the inside of the membrane is negative. And the number that we'll be talking about later on is minus 70 millivolts as a difference between the electrical conductivity of the inside to the outside. Now, when a nerve impulse is initiated, one of the first events that has to occur is called depolarization. Now, some stimulus is going to depolarize the neuron's membrane. Now, as we're looking at this picture here, we're looking at this from the aspect of this axon here, but this can also occur in the dendrites, cell body, but we're going to show this depolarization from here. And here is that polarized membrane here. When we just simply say there's a lot more sodium on the outside and more potassium on the inside. But as you depolarize it, a depolarized membrane, responding to some stimulus, is going to allow sodium to flow into the membrane, like we're trying to show on the second picture here. Something stimulates this membrane and depolarizes it, so sodium rushes in. Now, that stimulus can be a flash of light, a pinprick, anything that this membrane is responsive to, and it's going to cause sodium to flush in. And when sodium flushes in like this, we're going to exchange the charge. As more positive ions rush in, okay, the inside becomes much more positive. And that is what we call the action potential. That's going to be the initiation of it at that point. Now, this slide, we're going to take a look at how the action potential is actually propagated. Now, some impulses or some stimuli may be what we call subthreshold. Subthreshold means that it's not strong enough to generate the action potential, and the results of this depolarization fade out after a very short time. So in essence, you wouldn't detect these stimuli. Okay, but the action potential, once it starts, it's going to spread all across the entire length of the axon. Now, again, that may start, remember out here at the dendrites are the receivers. They'll have an action potential generator that will transfer through them, through the cell body, and then through the axon. And if it starts going along this axon, and this trigger zone right here called the axon hillock, if it's strong enough of a signal when it reaches that, it's going to travel all along the entire axon to the next nerve. Now, think of this as this is an example we call the all or none law. if the switch is flipped, it's going to go. Just like you can't turn a light switch on halfway and expect half the lights to come on. You know, you flip that light switch, they all come on or they all stay off. And the same is true here. Now, potassium ions, you know, that's the K ones, are going to rush out of the neuron after the sodium ions rush in. So what's happening here, if you look along here, here's our propagating action potential. The sodium ions are rushing in and they are diffusing across. And as they diffuse down this way, they depolarize the next segment, like we're showing right here. And as those ions diffuse further, they depolarize the next segment, and the action potential moves along. This is why the action potential goes along at its continued strength. It doesn't lose anything. Okay, now once you have this potential started, you've got to start repolarizing the membrane. Now at this point, back in here, which we call the refractory membrane, this membrane is trying to return to the action potential, return to the resting potential. It's unable to stimulate a response here, so if sodium ions diffuse this way, we're not going to generate an action potential going in this direction. It can only go in the direction that the membrane is in a polarized state. This is refractory, it's returning to polarized. Okay, it can only go to the excited one. Now, the way it returns to this resting potential is through the sodium-potassium pump. The sodium-potassium pump pumps the sodium ions out, potassium ions in, and returns the membrane to its polarized state. And remember from our study of membrane transport, this is an active transport mechanism, so ATP is burned in this process. Now, as the action potential moves along, if this occurs in the dendrite, it will move towards the cell body. So if the action potential is generated out here in the dendrites, it will travel towards the cell body, and then even in the axon, it will travel towards the next cell body. So think about these action potentials always moving towards some cell body. Now, I put it up here as a note here, we're going to show this in the next slide, that the impulses will always travel faster when there's a myelin sheath. But think about this. Here's why the myelin sheath, when you see the next slide, will make more sense. Right here is a depolarized membrane. Sodium has rushed in, flipped the membrane potential, and this is an excitable membrane over here, so these sodium ions have to diffuse into the next region, depolarize this little segment, and then they depolarize the next little segment, depolarize the next little segment, so it's going step by step. It's kind of like if you're walking up a flight of stairs and you touch every single step. Yeah, you'll get to the top of the stairs, but you'll get there slower than if you were to skip steps. And that's what we're going to look at on the next slide, is how the action potential can actually skip a few steps along the line. Now, movement along an axon that is myelinated is called saltatory conduction. And here's how this all works. At these segments right here, these nodes of Ranvier, here's the myelin sheath and the corresponding Schwann cell, here and here. This little gap between them is the only space where you have channels that are going to allow sodium ions to rush in. So if the impulse is traveling along this way here, moving along, and we flip the membrane potential here, so we allow sodium to rush in at this point, all right, this diffusing sodium does not have to depolarize all of this segment through here. All this part that I'm scanning along right now can be, the sodium ions diffuse, and no flipping of the membrane potential until it gets to here. Once those sodium ions get to here, they will open the voltage-gated channels, and sodium is going to rush in at this point. So instead of having sodium rush in at all the intermediate steps in through here, we can skip over this, kind of like skipping over, 2, maybe 3 stairs if you're trying to run up a flight of stairs. Yes, you'll get to the top of the stairs much faster, than you would if you hit every single stairs, just like the impulse is going to travel faster if it doesn't have to depolarize all these little intermediate pieces of the axonal membrane. Now those voltage-gated sodium channels are, aren't evenly distributed across this axon. For example, if you look along these segments along and through here that are covered by the Schwann cell and the myelin sheath, you'll have only about 25 per square micrometer of space along here. So they're pretty well spread out. But when you get to these nodes of Ranvier right here and here, the number of sodium voltage-gated channels here will jump up to like 12,000 per square micrometer in these nodes here. So they are going to be very, very receptive to any kind of influx of sodium. So if when the sodium reaches here, all those voltage-gated channels are going to jump open, just like you're seeing here, and see a huge influx of sodium to diffuse to the next one. And this is what we're showing right here. Here's where the membrane has been depolarized. So we got an action potential occurring here. This is an excitable membrane. So as the sodium diffuses all along and through here, and again, there's not very many voltage-gated channels all along and through here, but when they get to this segment, this node of Ranvier, where there's 12,000 plus, voltage-gated channels per micrometer, they easily depolarize. Now again, sodium is going to diffuse. This depolarization here, sodium is diffusing this way and this way. But if it tries to go back this way, this portion of the membrane is in a refractory period. It's resting. It's returning to its polarized state. So this one cannot be stimulated. So the impulse has no choice but to go this direction until it hits this node of Ranvier and becomes depolarized. So as you can see, the action potential is going to jump from one node to the next as it moves along, and the impulse transmission is going to be much faster than if we were trying to depolarize every single segment of the axon. All right, now this slide we're going to talk about what happens when the impulse finally reaches the next neuron. So in my picture here, imagine what's going on here. This neuron right here has received a signal. It's been stimulated, so along goes the action potential all along and through here, and it comes to the next one. Now, making this connection to the next one, you have to remember that neurons do not touch. The connection, maybe there's a better term to use, but they really don't touch. If you look at this enlargement right here, here is the first neuron. They got the first signal. We call that the presynaptic neuron, because this gap right here is called a synapse. Okay, a synapse is the point where one axonal terminal, transfers the impulse to the next neuron. The actual space is called the synaptic cleft. This whole area here is the synapse. And what happens is this. Along comes the action potential going down. When it gets to the axonal terminal, it's going to stimulate the release of a substance called a neurotransmitter. The neurotransmitter, and there's lots of different neurotransmitters, but the one we know the most about is called acetylcholine. Acetylcholine, let's just say that one here, acetylcholine gets released into this synaptic cleft. That's what these little green things are. When they come along here, they bind to the receptor proteins on the next neuron, or this is the postsynaptic neuron. The neurotransmitter binds, opens up a sodium gate, and in rushes sodium. So now sodium is going to be rushing into this postsynaptic neuron, or receiving neuron, and it's going to generate an action potential that moves all the way down again. Now, if the first one can transmit these neurotransmitters and stimulate the next neuron into an action potential, it's going to travel along at the same strength as the previous one. But the gap is the synaptic cleft. The whole union here between the presynaptic neuron and the postsynaptic neuron is called a synapse, and it's not very big. And once as I put down in here, once the sodium gates have been opened up here, the neurotransmitter, something usually happens to it. Either it's broken down, reabsorbed, released somehow, and it causes the gaps to close here, so the next neuron only fires one time. Okay, if the gaps were left open, you'd have a continuous flow of sodium in there. You'd have a continuous flow of action potentials constantly being generated.